Okay, so welcome to this next video in which we are discussing the cell cycle. Okay, so we're currently discussing the pathway downstream of growth factors. Okay, so we've discussed that growth factors, and the archetypal growth factor that we're using as an example is epidermal growth factor. They will activate receptors on the surface of cells. In the case of epidermal growth factor, you have epidermal growth factor receptor. This will then cause conformational changes in these growth factor receptors, which will lead to dimerization. Once they've dimerized, the tyrosine kinase domain on one of them will phosphorylate the tyrosine residues on the other, and the tyrosine kinase domain on the second one will then phosphorylate the tyrosine residues on the first. And by the way, I don't think I stressed this in the previous video, that process is known as autophosphorylation. Okay, so auto means self, and then self-phosphorylation. Now you might ask, well, why is it self-phosphorylation? Because it's not as though the tyrosine kinase domain on epidermal growth factor receptor 1 is phosphorylating its own tyrosine residues. It's phosphorylating the residues on the other one, so surely it should be cross-phosphorylation, and some people do call this cross-phosphorylation. Okay, but the reason it's called autophosphorylation is because you kind of view the dimer as though it's a single receptor now. And I think the reason that this sort of viewing the dimer as a single receptor has come about is because of the insulin receptor, where the insulin receptor is effectively like a growth factor receptor, but instead of having two two receptors which dimerize together, the insulin receptor is already dimerized effectively. So I think that's where the autophosphorylation comes from, and it's stuck for all of the growth factor receptors as well, even though they are dimers. Okay, right. So let's now look at the pathways downstream of the formation of these phosphotyrosine residues on the cytoplasmic side of our epidermal growth factor receptor. Okay, and as I said in the previous video, there are many different pathways that these can activate. We're going to study one of them to get the basic idea, and all the others will culminate in the same basic concept. Okay, so I hope that discussing one of them should be enough. Right, so the first thing that's going to happen is another protein is going to come and bind to these phosphotyrosine residues, okay, on the cytoplasmic side of our epidermal growth factor receptors. Okay, so let me just redraw out the picture that we've got so far. So here are our epidermal growth factor receptors dimerized together, like so, okay, and they're both sitting, of course, in the cell membrane. Then they will have epidermal growth factor molecules attached to them. So here in blue, here are the epidermal growth factors, okay. Here is the epidermal growth factor receptor molecule that spans the membrane. Okay, and of course we've got two of them, so I'll colour this one in red as well. And then on the cytoplasmic side, what you then have is this important tyrosine kinase domain here, okay, which has phosphorylated tyrosine residues on its opposite epidermal growth factor receptor. Okay, so here in vivid purple, this is the tyrosine kinase domain. Okay, and now let's show a phosphotyrosine residue here. So here is our tyrosine, and it's now got a phosphate group attached to it. Okay, so in vivid purple, this little ball here, this is going to represent our phosphate group coming off this tyrosine residue here. Now, what's going to happen is a protein is going to come and bind to phosphotyrosine residues on the cytoplasmic side of the epidermal growth factor receptor. Okay, and to do this, it has to have a special domain. So I'll show this here. So this is this protein, and it's got a special domain which is capable of binding to phosphotyrosine residues. And this special domain that it uses to bind to phosphotyrosine residues is known as an SH3 domain. Okay, and what this stands for is Sark homology 2. Okay, so S stands for Sark, H stands for homology, and then it's just domain 2, basically. Sark homology domain 2. Okay, right, so I'll colour in this SARC homology domain 2 in turquoise. Now this is basically a domain that you find showing up in a huge number of different proteins. So it's a conserved structure that you find in many different proteins, and the special feature that SH2 domains have is that they are capable of binding to phosphotyrosine residues, basically. Okay, so the SH2 domain will bind to this phosphotyrosine residue and recruit this protein to the phosphorylated underside of the epidermal growth factor receptor. Okay, so what is this protein known as? Well, it's known as GRB2. Okay, and this stands for growth factor receptor binding protein 2. Okay, so the G is for growth factor, the G is for growth factor, the R is then for receptor, okay, and then the B is for binding protein, and then 2, okay, and this growth factor receptor binding protein 2 then has another important domain that you find conserved in many different proteins. Okay, and this domain is known as an SH3 domain. So in blue here, this is another one of these conserved domains. Okay, an SH3 domain, and both of these domains were found in the sort of prototypical receptor tyrosine kinase enzyme, which is SARC. Okay, it's a very heavily studied one. So SARC homology 3 domain. So again, you find this sort of a domain in many different proteins. And what does an SH3 domain bind to? Well, it tends to bind to regions of a protein which are very proline rich. Okay, and this is going to be important because what the growth factor receptor binding protein 2, which I'll now highlight in yellow here, so we'll highlight this in yellow, what it's going to do is that it's effectively going to function as an adapter protein. Okay, so it's adapting the signal that is coming from the epidermal growth factor receptor, which is activated, and it's going to bind to the activated epidermal growth factor receptor, and it's then going to bind to the next molecule along, and it's just going to adapt the signal. It's going to bind to this so that another protein can then come and bind to it. Okay, so it uses this SH2 domain to bind to the epidermal growth factor receptor. It uses this SH3 domain to bind to the next protein along. Okay, and as I say, this SH3 domain is going to bind to proline-rich regions. So it likes to bind to proteins that have a lot of proline in. Okay, so I'll show you the structure of the amino acid proline then, and I'll do it down here. Okay, so proline is an interesting amino acid because its R group involves the amino group. So once again, I'll draw it as a residue. Okay, so I'll draw it as it would be within a protein. But this time it does not have a hydrogen coming off the amino group. Okay, so here is the alpha carbon in the middle with a hydrogen coming off, and here's the carboxylic acid group. Now, the R group then of proline consists of a 5-membered ring, effectively, well, not like that, a 5-membered ring that connects this carbon here to this nitrogen, and you have to put 3 carbons on to do this. So you'll have a methylene group here, a methylene group here, and then the final methylene group here. So this is a methylene group as well. And this then is the structure of the amino acid residue proline. Okay, so in turquoise there. So this is what SH3 domains like to bind to. They bind to regions of proteins where you have lots of proline. Okay, so what's then going to happen is that this SH3 domain is going to bind to a proline-rich region of the next protein along. So this little region that is bound to the SH3 domain, this will contain a lot of proline. So I'm highlighting it in pink here. And this is a proline-rich region. And for short, I'll abbreviate proline-rich region to PRR. P for proline, R for rich, and R for region. Okay, right, so what is this next protein along called? This next protein along is called SOS, okay, which has the rather fantastic name, Son of Sevenness, okay? So that's what SOS stands for. It stands for Son of Sevenness. Okay, and there are different son of sevenness proteins, but they seem to all do the same thing. So we'll just keep it general and just have a SOS protein there. Okay, so in turquoise here is the rest of our SOS protein. The whole thing, of course, is the SOS protein, but I'll colour this bit that isn't the protein-rich region in turquoise. Right, and son of sevenness is going to act as a guanine nucleotide exchange factor. It's going to activate G proteins. Okay, so the fancy term for something which activates G proteins is to call it a guanine nucleotide exchange factor. And then the abbreviation for guanine nucleotide exchange factor is usually to abbreviate it all down to GEF. So the G stands for guanine nucleotide, the E stands for exchange, and then the F stands for factor. So, SOS is what's known as a guanine nucleotide exchange factor. So what's going to happen is it's going to come up and bind to the growth factor receptor binding protein 2, and now it's going to be activating a G protein. Okay, so which G protein is it going to activate? Well, it's not going to activate a heterotrimeric G protein. It's not a G protein coupled receptor which activates on acts on heterotrimeric G proteins. Instead, it's going to work on a monomeric G protein. Now, monomeric G proteins only have one subunit, okay, but the essential principle is the same as for heterotrimeric G proteins, or in fact, you could say heterotrimeric G proteins are using the principle of a monomeric G protein. So the principle of a monomeric G protein is that it has two states, an off state, in which it has guanosine diphosphate, GDP, bound to it, and then an on state in which it has guanosine triphosphate, GTP, bound to it. Okay, and in the on state it will have all sorts of signaling roles, and in the off state it won't have these roles. Okay, and the idea of a guanine nucleotide exchange factor is that it's going to act on the monomeric G protein, rip the guanosine diphosphate off, and then bind a guanosine triphosphate on, and therefore it's going to turn the monomeric G protein on. Okay, so what is the monomeric G protein that SOS is going to activate? Well, it's going to be a Ras G protein. Okay, so there are many different Ras G proteins. Okay, there are 4 in total Ras monomeric G proteins. Okay, there is a NRAS G protein, okay, there is then a BRAS Ras G protein, and then there are 2 KRASs, okay, there is KRAS4A, and then there is also KRAS4B. Now, as far as we are concerned, all 4 of these are going to do the same thing. Okay, so we're not going to distinguish between the 4 different forms of Ras G proteins. Instead, we're just going to call our Ras G protein a Ras G protein, and we'll assume that that can be any one of these 4. Okay, so the next important thing to say about Ras G proteins is that they are attached to the underside of the phospholipid bilayer. So I've promoted the cell membrane up now. I was representing it by a single line. Now I'm going to represent it as 2 lines. Okay, I'm going to now represent the outer layer of phospholipids by 1 line, and then the inner layer of phospholipids by another line. Okay, so this is the inner leaflet of the phospholipid bilayer, which faces into the cytoplasm, and this layer that faces out into the extracellular fluid, this is known as the outer leaflet of the phospholipid bilayer. Okay, right. Now, Ras G proteins are going to be bound to the underside of the phospholipid bilayer. Okay, they're not integral membrane proteins, they're not in the membrane, okay, but they're going to be attached to the underside of it, they're going to be attached to the inner leaflet. Okay, now let's say initially that our Ras G protein is in the off state, and therefore has guanosine diphosphate bound to it here. Okay, so I'll colour it in here in orange. So this is our Ras G protein. So it's one of these 4 here, an NRAS, a BRAS, a KRAS4A, or a KRAS4B. Right, okay, now, how does the Ras G protein then get attached onto the underside of the phospholipid bilayer? Well, basically, it has a lipid moiety attached onto its side, which then implants into the inner leaflet of the phospholipid bilayer and holds the Ras monomeric G protein at the underside of the phospholipid bilayer. Okay, so what is this lipid moiety? Well, basically, it's a palmitoyl group. Now, what does it mean to have a palmitoyl group attached to you? Well, for that, I need to explain to you what the structure of palmitic acid is. Okay, so palmitic acid is the old name for a molecule that would now be called hexadecanoic acid. And although hexadecanoic acid is a bit more of a mouthful than palmitic acid, it's a more useful name because it tells you exactly what you're dealing with here. It tells us that we're dealing with a 16-carbon fully saturated carboxylic acid. So we'll start by drawing the carboxylic acid group here. Now we need 15 more carbons. We've got one here, we need 15 more. 14 of these are going to be in the form of methylene groups. Okay, so here's a methylene group. Now, I don't want to have to draw 14 of them out. So what I'm going to instead do is I'm going to bracket this methylene group like so. And I'm going to say, repeat this 14 times. And the way that you can do that is just bracket it around, put brackets around it, and then subscript it 14 to mean repeat this 14 times, basically. And then right on the end, you're going to then have a methyl group. Okay, so that will take us up to 16 carbons, 2 on either side, one in the form of this carboxylic acid group, the other in the form of the methyl group, and then the 14 in the middle in these methylene groups here. Okay, takes us up to a total of 16 carbons. Okay, so this is an extremely long hydrophobic tail that we have here. Okay, it does not interact well with water at all. What we effectively do to the Ras protein is we attach one of these palmitoyl groups here onto the side of the Ras protein, okay, onto one of its amino acid residues. Specifically, we attach it onto cysteine residues. Okay, and every Ras G protein gets one of these attached onto it. And it will sort of be sticking off the side of the Ras G protein. Now, it does not interact well at all with water. Instead, it will interact brilliantly with the other hydrophobic lipid groups that are in the hydrophobic core of the phospholipid bilayer. So what happens is this long hydrophobic tail here, which I'm representing by this line here, will implant into the phospholipid bilayer, and it will interact nice and stably with the other hydrophobic structures in that hydrophobic core of the phospholipid bilayer and hold the Ras monomeric G protein at the underside of the inner leaflet of the phospholipid bilayer. Okay, so that's how Ras G proteins are attached to the underside of the phospholipid bilayer. Okay, right. So next up, we need to discuss how these Ras G proteins are going to be activated then. Okay, we need to connect them back up to our growth factor receptor pathway here. So, we've now got our son of sevenness protein, our SOS protein here, bound and activated. And it's now going to activate the Ras monomeric G protein. So, it will come into contact with a Ras monomeric G protein, which is attached to the underside of the phospholipid bilayer, because now SOS is also attached to the underside of the phospholipid bilayer because it's attached to the GRB2, which is then attached to the epidermal growth factor receptor. It's a little bit more indirect, but it's still attached to the underside of the phospholipid bilayer. So SOS will now be able to act on the Ras G protein, and it will bind to it, and it will catalyze the conversion of the off-state Ras into the on-state Ras. Okay, so if I show this, I'll just draw the SOS here. Okay, so here is our SOS protein, okay, which will of course be bound to the SH3 domain of the GRB2. Okay, so here is SOS in turquoise here. In fact, I might actually draw the full cascade. It's a good revision. Okay, so here is SOS in turquoise. Then we have our proline-rich region in pink here. And then we have our growth factor receptor binding protein 2 here, with its special SH2 domain, which will bound to a phosphotyrosine residue on the underside of our epidermal growth factor receptor. Okay, so here in blue is our SH3 domain. Okay, the rest of GRB2 can be represented in yellow here. Okay, like so. And then we'll have the special SH2 domain here in turquoise, which is bound to a phosphotyrosine residue. So I'm now somehow going to have to squeeze my epidermal growth factor receptor in like here. Okay, so here we'll have our phosphotyrosine residue drawn like that. It's been demoted a little bit, and I'll just try and squeeze in the tyrosine kinase there. Okay, and they'll dimerized as well, which I've forgotten to show. Okay, so here's another epidermal growth factor receptor here, and they've shrunk a bit, and or the membrane's got bigger rather. Okay, and then here's our tyrosine kinase, like so. Okay, so let's colour those in as well. Oh, and I've forgotten to show the epidermal growth factor bound to these as well. But here we go. Here are the epidermal growth factor receptors dimerized together with the epidermal growth factor bound to them. Here, 1, 2, both of them will have epidermal growth factor bound. So I'll colour these in blue. And then we'll have our tyrosine kinase domains down here. Okay, and then we've got that phosphorylated tyrosine there that is bound to the SH2 domain of our GRB2 here. Right, okay, so what will happen is the son of 7-less protein will bind to the monomeric Ras G protein that is in the off state, and what will now happen is that will allow the Ras G protein to give up its guanosine diphosphate. So before it bound to the SOS G protein, it was incredibly strongly bound to that guanosine diphosphate. It was extremely unlikely that it would ever let go of it and spontaneously activate itself. But once it binds to the SOS protein, what that does is it weakens the bond between the Ras and the guanosine diphosphate. The guanosine diphosphate can then leave the Ras, and what will replace it? Well, there's a much higher concentration within the cell of guanosine triphosphate than guanosine diphosphate, so it's far more likely that a guanosine triphosphate molecule will come in here to replace the GDP than a GDP will come back in, basically. Okay, then the Ras in the on state will now go off and activate the next protein along in this great cascade. Okay, so we've now got our Ras G protein in the on state. Okay, so once again, here is the phospholipid bi there. Here is our Ras G protein underneath it, and the SOS G protein exchange factor, okay, has now activated the Ras G protein, so it's got GTP bound. And once it's got GTP bound, it will cleave away from the SOS, okay, so it will be now floating freely underneath the inner leaflet of the phospholipid bi there, still attached via this lipid moiety that it's got stuck on the side of it. Okay, so we now want to see what is our Ras GTP subunit here going to go, well, monomeric G protein rather. I'm thinking of heterotrimeric G proteins, calling it a subunit. It's a protein in its own right. What is this now going to go off and activate? Well, it's going to activate an enzyme known as RAF. Okay, and again, there are multiple RAF enzymes. Okay, so we'll discuss RAF enzymes. They are kinase enzymes. Okay, so they phosphorylate serine and threonine residues majorly. Okay, so RAF kinases. So you have ARAF, BRAF, and CRAF in humans. And what's going to happen is the Ras monomeric G protein here in the on state is going to activate these RAF kinase enzymes. Now again, for our purposes, we are going to view all three of these RAF kinases as equivalent effectively. So we're just going to call it RAF, okay? And it's going to be in the cytoplasm. So we'll have our RAF kinase shown here in the cytoplasm. Okay, and what happens is, this isn't fully understood, but the Ras monomeric G protein, which is in the on state, is going to lead to the phosphorylation of a tyrosine residue within our RAF kinase. Okay, so here is a tyrosine residue within our RAF kinase, and basically it gets phosphorylated. Okay, and it's not fully understood how this happens. But what is known is that the RAF kinase enzyme comes over, interacts with the Ras monomeric G protein, and then gets its tyrosine residue phosphorylated, but it's not understood how. We don't think that the Ras directly has a tyrosine kinase within it. We think instead it probably recruits another enzyme, which is a tyrosine kinase, which then phosphorylates the RAF kinase. But that's what happens. The RAF kinase gets phosphorylated on a tyrosine residue when it interacts with a Ras monomeric G protein, and this turns it from being in an inactive state to being in an active state. So it's now going to go off and phosphorylate the next protein along in this signaling cascade, and we'll come back to that in the next video.